Most people believe the nutrients that protect life the most are the ones they hear about constantly: vitamin D, omega, 3S, vitamin C, magnesium. But a long-running scientific investigation discovered something unexpected, something hardly anyone talks about anymore. A study published in the American Journal of Clinical Nutrition followed 829 people over 2 decades and measured 146 different nutrients in their diets. Not a handful. 146 separate compounds were tracked through structured dietary interviews every 5 years, and deaths were confirmed through medical records, certificates, and autopsies. At the end of those 20 years, researchers asked a simple but powerful question: which nutrient showed the strongest link to living longer? It wasn't the nutrients that dominate health headlines. It wasn't vitamin D, omega-3 fats, or vitamin C. It wasn't magnesium either. The nutrient that showed the strongest inverse association with dying from any cause was a compound most physicians have never mentioned to their patients. And here's what makes the finding especially important. This compound appears in very high concentrations in a food that very few people eat today. Even more significant, levels of it inside the body begin dropping sharply after the age of 60, precisely when the body may need it most. I'm Dr. Becker, and every week I spend a significant amount of time reviewing primary research papers, not summaries, not headlines, and not press releases. The reason is simple. There is often a wide gap between what the scientific literature actually shows and what eventually reaches the public conversation. What I'm sharing today comes directly from peer-reviewed research published in journals like Nature Cell Biology, Cell Discovery, Frontiers in Public Health, and a 2024 study conducted at the University of Basel. These are respected scientific publications, and the work behind them involves teams of researchers from multiple institutions. Before going further, a quick but important note. Everything we discuss today is educational and based on published research. Some of the topics, particularly fasting approaches and certain foods, can interact with medications or health conditions. If you're taking anticoagulants, managing diabetes with insulin or sulfonylureas, or dealing with active cardiovascular disease, you should speak with your physician before making changes. As we move forward, I'll point out where those cautions matter most. The compound at the center of this discussion is called spermidine. And from the beginning, I want to be clear about 3 things: what the research confirms, what scientists strongly suspect, and what still needs to be proven. The truthful version of the story is actually more interesting than exaggerated claims you sometimes hear online. Today I'm going to walk through 5 key studies that together create a surprisingly consistent picture. By the end, you'll understand what spermidine is, where it comes from, what happens inside cells when levels decline, and why a 2024 paper in Nature Cell Biology changed how researchers think about intermittent fasting. We'll also look at recent research involving muscle stem cells, including its limitations, and finally we'll talk about what a realistic, evidence-based dietary strategy actually looks like. If you find these deep dives into real science helpful, subscribing helps support the work behind them. No pressure. I simply appreciate you being here and engaging with the research. To understand why spermidine matters, we need to begin with epidemiology, the long-term population studies that reveal patterns over time. One of the most important investigations in this field is the Bruneck study, a cardiovascular cohort project that began in northern Italy in 1990. Since then, researchers have continued examining the same population through repeated health assessments for more than 3 decades. In a 2018 analysis published in the American Journal of Clinical Nutrition, scientists examined data from 829 participants between the ages of 45 and 84. The follow-up period extended across 20 years. Instead of relying on simple food diaries, trained dietitians conducted structured interviews every 5 years to assess dietary patterns in detail. Causes of death were confirmed using medical documentation and autopsy reports, ensuring a high level of accuracy. The researchers analyzed 146 dietary compounds and evaluated how each one related to mortality risk. Among all the nutrients measured, spermidine stood out. Participants with the highest intake levels showed a markedly lower risk of dying from any cause compared with those consuming the least. In fact, the difference between groups was striking. Individuals in the highest third of spermidine intake had a 20-year cumulative mortality incidence of 0.38, while the lowest third reached 0.48. When translated into raw numbers, that meant roughly 15.1 deaths per 1,000 person years in the high intake group, compared with 40.5 inches the low intake group. That is not a small difference, and the pattern wasn't isolated to one population. Researchers replicated similar findings in an independent Austrian cohort called SAFIR, which included 1,770 participants aged 39 to 67. The protective association appeared especially strong when it came to cardiovascular disease. Before we move further, it's important to acknowledge a limitation that researchers themselves emphasize. Studies like this are observational. They can identify associations but cannot prove direct causation. It's possible that people who consume more spermidine-rich foods also follow broader dietary habits that contribute to better health outcomes. Scientists account for many of these factors statistically, but residual confounding can never be completely ruled out. Even with that caveat, the consistency across studies makes the finding difficult to ignore. And when researchers began examining US data, they saw something very similar emerging. The US data comes from a 2022 analysis published in Frontiers in Public Health using the NHANES database, the National Health and Nutrition Examination Survey. Researchers followed 23,894 American adults from 2003 to 2014. During this period, 2,365 deaths were documented, including 736 from cardiovascular causes. Using Cox proportional hazards regression models, the team found that participants with the highest dietary spermidine intake had about a 30% lower risk of dying from any cause and a 32% lower risk of cardiovascular death. These results held even after adjusting for age, sex, physical activity, total caloric intake, and other lifestyle factors. What makes this compelling is that it was observed in a heterogeneous, diverse population, far from the relatively homogeneous cohorts in Italy or Austria, yet the association remained consistent. So, what makes spermidine more than just a nutritional correlation? We now have plausible biological mechanisms explaining why it might impact longevity, and they converge on one of the most studied cellular processes in aging research: autophagy. Autophagy, derived from the Greek for self-eating, is the process by which cells identify damaged components, misfolded proteins, dysfunctional mitochondria, and other debris, wrap them in double-membrane vesicles called autophagosomes, and deliver them to lysosomes for breakdown and recycling. Think of it like your body's internal cleaning crew. As your cells carry out metabolism, they generate byproducts and accumulate damage, similar to how a kitchen accumulates dirty dishes, worn utensils, and packaging. Autophagy is the team that comes through and removes what's no longer functional, ensuring that cellular processes remain efficient. The challenge is that autophagic efficiency declines with age, right when cellular damage begins to accumulate more rapidly. Proteins associated with neurodegeneration, damaged mitochondria producing inflammatory signals, and dysfunctional organelles start to build up. Aging, in many ways, resembles a gradual accumulation of cellular debris, and autophagy is the cleanup system designed to prevent that buildup. Spermidine activates autophagy through at least two well-understood mechanisms. One involves inhibiting histone acetyltransferase activity, an epigenetic process that upregulates genes responsible for autophagy initiation. The other activates AMPK, a cellular energy sensor that triggers autophagy-related proteins such as LC3, Beclin-1, and P62. These pathways are not theoretical. They've been observed in multiple experimental systems. Yoshinori Osumi received the Nobel Prize in Physiology or Medicine in 2016 for his foundational work in autophagy mechanisms, providing the core framework for understanding these processes. The 2024 research brings a critical twist to this story. While previous studies established that spermidine activates autophagy, Sebastian Hofer and colleagues asked a deeper question: is spermidine necessary for autophagy, or can the cellular cleanup process occur without it? Their study, published in Nature Cell Biology in September 2024, involved over 40 researchers from 20 institutions across multiple countries. The team examined the interplay between fasting, spermidine, and autophagy in yeast, nematodes, fruit flies, mice, and humans. Across all these species, disrupting the spermidine EIF5A axis eliminated the anti-aging benefits typically associated with fasting. To visualize this, imagine EIF5A as a car and spermidine as the key. Fasting acts as the signal to turn the key, but without spermidine, the engine autophagy cannot run, regardless of how much fuel, metabolic energy is present. For people over 60 whose baseline spermidine levels are lower than at age 30, this has practical implications. Intermittent fasting may still signal the body to initiate autophagy, but if spermidine is insufficient, the process might be slower or less effective. It's essential to clarify the limitations here. The longevity effects were demonstrated in model organisms, not humans. We don't yet have a large-scale clinical trial showing that higher spermidine levels extend human lifespan. What we do have is a mechanistic understanding that spermidine is critical for autophagy and epidemiological evidence linking higher dietary intake to lower mortality. Together, these findings create a coherent picture, though the direct causal link in humans remains to be proven. Spermidine's influence extends beyond autophagy. Consider mitochondria, the energy-generating organelles within cells. After age 50, the number of functional mitochondria decreases substantially, with some tissues showing a 30–50% reduction by the seventh decade. Dysfunctional mitochondria not only produce less ATP, the energy currency of cells, but also generate excess reactive oxygen species, damaging nearby proteins, lipids, and DNA while triggering inflammatory pathways. A December 2024 study from the University of Basel used human-induced pluripotent stem cell-derived neurons to examine spermidine's impact on mitochondrial health. The study found that spermidine increased ATP production and mitochondrial membrane potential while reducing reactive oxygen species in both young and aged neurons. Spermidine also improved maximal respiratory capacity, effectively boosting the performance of healthy mitochondria while facilitating the removal of damaged ones. This process, called mitophagy, relies on the PINK1 pathway, which selectively flags dysfunctional mitochondria for recycling. In simple terms, spermidine helps cells take out malfunctioning batteries while optimizing the ones that still work. Another critical mechanism relates to chronic, low-grade inflammation, or inflammaging, which becomes more pronounced after age 50. Unlike acute inflammation, which is a necessary response to injury or infection, inflammaging is persistent background inflammation that contributes to cardiovascular disease, metabolic dysfunction, neurodegeneration, and cancer. By the mid-60s, most adults show elevated levels of inflammatory markers like C-reactive protein, interleukin-6, and TNF-alpha, even without diagnosed disease. Spermidine counters this process through several pathways: epigenetic inhibition of EP300, modulation of NF-kappa B signaling, and activation of autophagy and mitophagy to remove cellular debris that triggers inflammatory signals. Hofer's 2024 study demonstrated that fasting's anti-inflammatory effects in aged animal models depended entirely on a functioning spermidine, EIF5A axis. These mechanisms converge in ways that matter profoundly for aging. Inflammaging impairs insulin signaling, contributes to atherosclerosis, promotes neuroinflammation, and creates an environment hostile to stem cell-mediated repair and regeneration. Spermidine, through its multiple effects on autophagy, mitochondria, and inflammation, addresses each of these age-related pathways simultaneously, making it a uniquely promising target for longevity research. The fourth area of research into spermidine involves muscle stem cells, which is particularly relevant as we age. Skeletal muscle contains a population of dormant stem cells called satellite cells. These cells sit quietly within muscle tissue, waiting for the signal to repair or regenerate fibers following injury or exercise. Their activity is essential not only for recovery after trauma, but also for adaptation to resistance training. Unfortunately, both the number and functional capacity of satellite cells decline with age, contributing to slower muscle repair and reduced adaptation to exercise in people over 50. A September 2024 study published in Cell Discovery, a Nature Publishing Group journal, investigated spermidine's role in satellite cell activation. Researchers compared young mice 6 months old with aged mice 30 months old. Using metabolomic analyses of isolated satellite cells, they observed significantly lower spermidine levels and reduced spermidine to spermine ratios in older animals. Furthermore, genes encoding enzymes critical for polyamine biosynthesis were downregulated in aged satellite cells, suggesting a mechanistic link between reduced polyamine availability and diminished regenerative capacity. The team then restored spermidine levels in both young and old mice, observing a marked improvement in satellite cell activation. Mechanistically, this effect was traced to EIF5, hypocyanation, which regulates translation of MyoD, the master transcription factor for muscle differentiation. This aligns with the broader understanding of spermidine as a key substrate for cellular processes, in this case allowing satellite cells to effectively repair and grow muscle tissue. It's critical to emphasize, however, that this is mouse data. No randomized controlled trials have yet demonstrated that spermidine supplementation in humans improves satellite cell function or increases muscle mass. The mechanisms are plausible and consistent with other research on spermidine, but we cannot yet confirm these effects occur in people at the magnitude suggested in animal studies. Nonetheless, sarcopenia, the progressive loss of muscle mass and function, affects 10–15% of people in their 60s and up to 50% by their 80s. Understanding potential cellular contributors, such as polyamine decline, is valuable in framing preventive strategies, even if definitive human evidence is pending. A common question arises when discussing spermidine and cell proliferation. Does it raise cancer risk? Polyamines, including spermidine, are necessary for cell proliferation, which theoretically could support tumor growth. Early research raised concerns about supplementation in oncology contexts. However, epidemiological data, including the Brunk study, does not show an increase in cancer incidence associated with higher dietary spermidine intake. In fact, some data suggest protective associations for cancer mortality. For individuals with active cancer or undergoing treatment, any dietary changes should be discussed with an oncologist before introducing spermidine-rich foods or supplements. Moving from cellular mechanisms to practical application, the foundation of a spermidine-oriented dietary strategy is food, not supplements. This distinction matters because the body tightly regulates circulating spermidine levels. A 2023 study examining purified spermidine supplementation at 40 milligrams per day for 28 days in older men found no significant increase in serum or urinary spermidine concentrations. While local effects in tissues or the gut cannot be ruled out, this finding highlights that increasing dietary intake is a more reliable way to influence spermidine biology. The richest known food source of spermidine is wheat germ, with concentrations around 2,437 nanomoles per gram, substantially higher than most commonly consumed foods. A tablespoon of wheat germ provides roughly 1–2 milligrams of spermidine, meaning 1 to 2 tablespoons daily added to yogurt, oatmeal, smoothies, or salads can achieve meaningful intake. It is affordable, widely available, and aligns with broader dietary patterns associated with longevity. Contraindications include celiac disease or wheat allergy, in which case wheat germ should be completely avoided. Other dietary sources include natto, a Japanese fermented soybean product. Natto is exceptionally high in spermidine and also provides vitamin K2. Its strong fermented flavor and sticky texture may be off-putting to those unfamiliar with it, but it represents a nutritionally potent option for those willing to acquire the taste. Additional contributors are shiitake mushrooms, mature cheeses, broccoli, cauliflower, green peas, and spinach. These foods align with the Mediterranean dietary pattern, which has been repeatedly associated with lower cardiovascular risk and longevity. A 2023 analysis specifically examining cardiovascular mortality found that vegetables, legumes, cereals, nuts, and certain cheeses, the primary sources of dietary spermidine, were the strongest predictors of protection, suggesting that part of the Mediterranean diet's benefits may operate through polyamine biology. Intermittent fasting intersects with spermidine biology in interesting ways. The 2024 Nature Cell Biology study demonstrates that fasting increases endogenous spermidine, which drives EIF5A hypocyanation and activates autophagy. Dietary spermidine provides exogenous substrate, while fasting stimulates biosynthesis. These pathways are complementary rather than redundant. For most adults over 50 without diabetes, pharmacotherapy, a 12-16-hour fasting window finishing dinner around 7-8 p.m., and consuming breakfast at 8-9 a.m. is supported by evidence for metabolic and longevity benefits. Individuals on glucose-lowering medications require physician guidance before implementing fasting protocols. Resistance exercise also synergizes with spermidine mechanisms. While direct spermidine data are lacking, exercise activates AMPK, the same energy-sensing pathway influenced by spermidine, and stimulates satellite cell activity while reducing systemic inflammation. Engaging in 2 to 3 sessions per week, targeting major muscle groups, legs, back, shoulders, chest, paired with adequate protein intake, complements dietary and fasting strategies. Resistance training remains one of the most robust interventions for sarcopenia prevention in older adults. Finally, ongoing clinical trials are exploring spermidine supplementation. The most notable is the PolyCAD trial, enrolling 187 participants aged 65 plus with coronary artery disease, randomized to 24 milligrams per day of spermidine or placebo for 48 weeks. Primary outcomes include cardiac remodeling, exercise capacity, muscle mass, and systemic inflammation, with results expected in 2026. Smaller trials investigating cognitive outcomes show mixed results, highlighting the importance of dosage, baseline spermidine status, and population differences. Until these trials are complete, focusing on food sources remains the most evidence-supported approach. Having explored the mechanistic science, the epidemiology, and the experimental animal data, the final piece of the puzzle is translating these insights into a practical, evidence-based approach while being transparent about limitations and safety considerations. What does this mean for real life, particularly for adults over 50 who are concerned with longevity, cardiovascular health, muscle preservation, and cognitive resilience? First, let's address supplementation. The data on purified spermidine supplements remain preliminary. Studies in older adults have shown that daily doses of 40 milligrams are safe, with no major adverse effects, but crucially, serum and urinary spermidine levels did not reliably increase. This suggests that the body maintains tight homeostatic control over circulating spermidine. Local tissue effects may exist, but the systemic impact is less predictable than with dietary intake. The most important takeaway here is that supplements are not a shortcut. They are a research tool and, for now, should be considered experimental outside of clinical trials. The ongoing PolyCAD study may provide more clarity in 2026, but until then, the safest and most evidence-supported approach is diet first. Wheat germ emerges as the cornerstone of a spermidine-focused diet. With approximately 2,437 nanomoles of spermidine per gram, even modest daily servings 1 to 2 tablespoons deliver meaningful intake. Incorporating wheat germ into breakfast routines, whether stirred into yogurt, blended into smoothies, added to oatmeal, or sprinkled on salads, is practical and cost-effective. Importantly, this approach aligns with broader dietary patterns, particularly the Mediterranean diet, which is rich in vegetables, legumes, whole grains, nuts, and certain cheeses, the very foods most strongly associated with cardiovascular protection in spermidine-related analyses. For those with celiac disease or wheat allergy, wheat germ is off limits, but other foods like natto, shiitake mushrooms, and spinach can contribute, albeit at lower concentrations. Intermittent fasting can complement dietary intake. Mechanistic studies, particularly the 2024 Nature Cell Biology research, show that fasting elevates endogenous spermidine production, activating EIF5, a hypocyanation, and autophagy. A practical fasting window for most adults over 50 is 12-16 hours, typically finishing dinner around 7-8 p.m. and consuming breakfast between 8-9 a.m. This practice not only enhances spermidine biosynthesis but also provides additional metabolic benefits, including improved insulin sensitivity and cellular repair processes. For those on glucose-lowering medications or with active cardiovascular conditions, fasting should be implemented only under medical supervision. Resistance training is another synergistic component. While direct human data linking spermidine intake to muscle mass are still forthcoming, mechanistic overlap suggests additive benefits. Exercise activates AMPK, the same energy-sensing pathway that spermidine influences, stimulates satellite cell activation, and reduces systemic inflammation. 2 to 3 resistance sessions per week, targeting major muscle groups, paired with adequate protein intake, is highly recommended. This approach addresses sarcopenia risk, enhances mitochondrial function, and supports overall metabolic health. Safety considerations are essential when translating spermidine science into practice. For those on anticoagulants like warfarin, wheat germ's high vitamin K content can affect INR levels, necessitating monitoring rather than elimination. Individuals experiencing gastrointestinal distress such as bloating, diarrhea, or blood in stool after introducing wheat germ should be evaluated for celiac disease or wheat intolerance. High-fiber foods can interfere with absorption of some oral medications, so spacing intake by at least 2 hours is prudent. For people on medications like metformin or statins, mechanistic overlaps exist, though no human studies confirm interactions. It's wise to discuss any significant dietary changes with a prescribing physician. Importantly, the timeline for meaningful effects is long-term. Epidemiological studies that demonstrated reduced mortality tracked dietary patterns over 20 years. Cellular changes, improved autophagy, mitochondrial efficiency, and reduced inflammaging occur continuously, but the clinically meaningful outcomes, cardiovascular protection, preserved cognitive function, and maintained muscle mass emerge over years, not weeks. Think of spermidine-focused dietary patterns as a permanent lifestyle choice, rather than a quick intervention. Consistency matters far more than short-term optimization. Integrating all of this, a practical framework for adults over 50 looks like this. Build your diet around the Mediterranean pattern, ensure a daily serving of wheat, germ, or other spermidine-rich foods, consider intermittent fasting if medically appropriate, and engage in regular resistance training. This combination addresses multiple aging mechanisms: cellular clearance through autophagy, mitochondrial health, inflammaging, and muscle stem cell activity using interventions that are evidence-based, low-risk, and complementary. Finally, let's reflect on the intellectual takeaways. Spermidine is not a miracle or a single-cause solution, but it is a well-characterized molecular substrate that connects multiple longevity pathways. Epidemiological data in European and American populations show that higher dietary intake is linked to lower all-cause and cardiovascular mortality. Mechanistic studies explain why. It drives EIF5, a hypocyanation, activates autophagy, protects mitochondria, and reduces low-grade chronic inflammation. Animal studies provide compelling evidence for muscle stem cell benefits, and early human trials show promise, albeit inconsistently. The cumulative story is coherent and actionable. Dietary spermidine intake, combined with lifestyle practices like fasting and resistance training, constitutes a practical, mechanistically informed approach to supporting health as we age. The beauty of this science is its honesty. We know where the evidence is strong and where gaps remain. The decline in spermidine levels after 60 is not a fate sentence, it is information. By adopting consistent, science-aligned dietary and lifestyle patterns, you can slow or mitigate downstream consequences of this decline. Wheat germ, the Mediterranean diet, intermittent fasting, and exercise are tools you can use today with a high probability of benefit and minimal risk. In closing, spermidine research represents a rare intersection of epidemiology, cellular biology, and practical nutrition. It demonstrates how a single compound, often overlooked, can illuminate the underlying biology of aging and longevity. The take-home message is simple. Focus on consistent, long-term dietary patterns, integrate complementary lifestyle strategies, and engage with the science honestly. This is not about quick fixes, it's about building the foundation for healthy aging. Dr. Becker's evidence-based approach shows that with thoughtful, informed choices, the decline in critical nutrients like spermidine doesn't have to dictate the trajectory of aging. Instead, it can be managed, optimized, and leveraged to enhance cellular function, cardiovascular resilience, cognitive health, and muscular vitality for decades to come.